Hello, welcome everyone, good afternoon. Welcome to this session of the Alumni Festival. My name is Helena Knowles and I'm a university lecturer and a Royal Society University Research Fellow. And I will be talking to you today about some research in my lab on MRI at the nanometer scale. So maybe a bit of background to start with. So I did my PhD at the University of Cambridge in 2011 I started, and then I was a research fellow at St. John's for a few years before going to Harvard for a postdoc of a couple of years. And I've just returned last year to start my own lab here in Cambridge. So a couple of sort of housekeeping notes to begin with. So your microphones and video cameras will be turned off during this session, but we would like to make it as interactive as possible. So please do type in any questions that you have into the Q&A box. And if I can see them during the talk, I will try and, if possible, answer them as we go along. But there will definitely be a Q&A session at the end where you can ask lots of questions. But anything that comes up during the talk, do type it in and I will try and address it on the spot. As well, this session is being recorded and you'll be able to watch it again if you so wish. It'll be online on the university's website from October onwards. All right, so let's get started. Okay, so I'm imagining that most of you are familiar with magnetic resonance imaging techniques. At least you've heard of MRI, for instance, probably, which is typically used in hospitals in a clinical setting to image tissue. So here you can see on the left a typical MRI image, and the spatial resolution here is on the order of 1 millimeter, maybe slightly less. So that's a really nice diagnostic tool. On the other hand, there's a very similar technique called nuclear magnetic resonance, NMR, which allows you to look at the structure of molecules, of single molecules, which is really powerful. So you can take proteins and you can find out what they look like. But you do need a very large number of identical copies to get this information. So you need billions and billions of identical molecules to get enough signal to noise to be able to resolve these structures. So this is a really nice technique because it's so non-invasive that you can put a living human being into an MRI machine and they're perfectly happy and nothing bad happens. You get these amazing images. So it's a really good technique in principle, but it can't be, at the moment, the techniques that we have don't allow us to really scale this down to smaller length scales. So say to nanometers, which is the scale of single molecules or the scale of sort of subcellular compartments. So if you really want to do nanoscale imaging, single molecule imaging, you kind of need a new technique. And just maybe as a reminder of how big or how small a nanometer is, I quite like this analogy of taking the Earth and squeezing it down to the size of an orange. And if you apply that same process again to the orange and you squeeze it down by the same amount, you get to a nanometer. So it's really very, very small. So one in a billion, 1 to a billion ratio. So that's how small we're talking, really getting to the single molecule size. So both of these techniques, the MRI and the NMR, are based on imaging spins, looking at spins in our body. So typically proton spins that are ubiquitous, they're in water, they're pretty much everywhere, and from their density we can get these nice images in MRI. And so one question that you might have is what is even a spin? And it's a slightly sort of mysterious quantum object. And it's, we call it an intrinsic magnetic moment of a particle. And it's not actually physically spinning, but it has many analogies with a spinning object, which is why it's called a spin. And from a sort of mathematical treatment point of view, it can be treated in a similar way to a spinning object. So you just have to imagine them as tiny little compass needles that are associated with single particles such as electrons, but also the nuclei of atoms all around us. And so we've seen how this technique is really nice because it's non-invasive, And so it won't kill whatever, if you're trying to image something that's alive, it won't kill it. So it's a really nice technique. So how are we going to scale this down? Well, there are new technologies that are based on quantum mechanics that could allow this. And the technique that I'm going to talk about today is based on impurities in diamond. And again, you're probably familiar with some aspects of diamond, in particular as jewellery, like shown here. Typically, most diamond jewellery is actually transparent and colourless diamond, but then there are these sort of so-called fancy, colourful diamonds that are also very interesting from a quantum technologies perspective. So these colours come from impurities or inclusions in the diamond, and we will look at what they do in the next few slides. So this is just looking at a diamond lattice. You can see the carbon atoms here, and they're arranged in this particular very tight structure, the diamond lattice. And the defect that we're interested in from a quantum sensing perspective is a nitrogen atom next to a vacant site. So what happens is you take a carbon atom out of the diamond lattice, and next to it, you leave an empty site, and this together forms the nitrogen vacancy center. And this indeed gives a color that's sort of pinkish. So pink diamonds will be full of these nitrogen vacancy centers, or NV centers as we call them. And around this defect, this center, there will be a spin, which is associated with the electrons that are sort of bound to this defect. So they will create this spin. And of course, now we have a little magnetic moment, this spin, that can interact with other spins in its vicinity. And that will be the basis for the quantum sensing experiments that I'll show you later. But maybe to take a step back, we can ask ourselves, well, how do these NV centers make quantum sensors, but perhaps also, what even is a quantum sensor? So you're actually all familiar with quantum mechanics processes in sort of daily daily devices that you would be using, for instance, in your mobile phones. The rules of quantum mechanics are needed to describe how any kind of semiconductor technology works. So any kind of electronics has quantum mechanics built into it. Lasers, of course, as well, atomic clocks used in GPSs and LEDs as well. So there's already a lot of sort of quantum technology around us. It's just nobody really calls it quantum technology because we're actually looking at something slightly different. So just because it has quantum mechanics in it doesn't really make it a quantum technology. What we want to see is something that's making use of some of the slightly stranger properties of quantum mechanics, such as entanglement and superposition of states, to give us an enhanced performance over conventional technologies. So today we're not going to be looking at these technologies, which are inherently quantum, but that's not what we're talking about when we say quantum sensors at the moment. So this little spin that's trapped in the diamond, it can be in two different states. We'll call them down and up, as you can see here, the arrows pointing up and down. And you can see the energy axis here. So the down state is lower in energy than the up state, and this is because of the geometry of the crystal in this case. But the important thing is that these are discrete energies. So the energy is quantized. There are no energies in between. We have these two energy levels for the two spin states. And now, because they behave like little magnets, if we bring a big magnet in their vicinity, these energy levels will move up and down. So again, a little bit like a compass needle wiggling around, say, in the Earth's magnetic field. And of course, this doesn't have to be a large magnet, as I showed in the previous picture, but it could be another spin. This could be the magnetic field originating from a different spin that comes close to this nitrogen vacancy spin. Or it could be a collection of spins trapped in a protein or another large molecule that we're interested in. And so now, as I mentioned earlier, we want to make use of this system and control it in such a way that we can create these strange superposition states. I'm sure you've heard of this Schrodinger's cat thought experiment, where a cat is at the same time alive and dead, which sounds nonsensical, but this is indeed how quantum mechanics works. And we can have these states where this spin is not just in state up or down, but is actually in a superposition state of both states. And we only find out which state it's in after we measure it. So we're going to look at how we control this in the lab, and I will show you some pictures of what this looks like in the lab in a moment. But again, a sort of simplified diagram of how the system works is that we have these two levels, and they're this spin and a diamond, and now we have sort of two handles from quantum optics that we can use to control this spin. So we have a laser that emits green photons, and so single light particles in the visible green wavelength, and then we have microwaves as well, which are resonant with the energy difference between these two spin states. So what happens if we now shine this green laser onto the spin, onto this defect? We can see that if it's in the lower energy state, if it's pointing down, it emits a red photon. The center gets excited, and as it decays, it emits in the red. So we can collect those red light particles, those red photons, with using very sensitive detectors, and we can know that if we detect them, we were in the down state. Because if we're in the up state, actually what happens, this is due to the properties of this defect, is that we can excite the defect when it's in the up state, but when it decays, it actually doesn't emit any photons. So we can read out this quantum state of the defect purely using optics. And then the other sort of control knob that we have is these microwaves. So we can apply microwave pulses. We use microwave antennas to coherently drive this spin from its ground state to the upper state. So again, here illustrated with these arrows. We can also use slightly shorter pulses to create these kinds of superposition states. So we really have full control over the quantum state of this defect. Yes, so superposition states as shown down here on the left. And now in the lab, this is how it looks. So here in the middle, you see a big, well, at least large-ish millimeter-sized chunk of diamond. And you can see that it's glowing red. So we're exciting from below. We have a green laser at 532 nanometers. And we're exciting this piece of diamond, which is very dense in defects. And so you can see these red photons that I mentioned earlier from the spin down lower energy level state. As we continuously excite, they keep emitting these red photons. You can see that in the glow here. And now, thanks to this readout and this preparation control that we have using the microwaves, we can now generate such a superposition state. And then we can track it over time. So we can let it evolve for a little bit and then measure it and see whether it was in up or down state. And we can see that it's processing over time, as we would expect, in an external magnetic field. So this is a sort of typical nice time evolution of a spin in its environment. And this is a sort of very nice unperturbed environment with regular precession frequency. But of course, if there are interactions with other spins in its vicinity, this would look slightly different. And that's what gives us the hint of what's happening in the environment. And this really being able to create these superposition states and letting them evolve, these states are very fragile, but they can also be very sensitive. Because they're so fragile, they're very sensitive to their environment. And this is what gives us the sort of quantum benefit that makes the NV center a quantum sensor and gives us the enhanced sensitivity to try and detect single spins in its nanoscale vicinity. So, for example, we can tailor these microwave pulses in such a way that we become sensitive to other electronic spins in the diamond crystal that are nearby. So you can see these resonances here in the plot below at different frequencies. And this we're able to acquire just using a single defect. So it's really the sort of nanoscale environment of this defect. And we can look and see what other spins surround it. And in this case, this is nitrogen spins in its surrounding. And we can even take this one step further and try and see, can we actually spatially localize them? So here on the left, you can see this it's like a 2D projection of a diamond lattice. So all of these little gray dots are possible sites for the defect. They are the lattice sites of the carbon atoms in the diamond lattice, and we've done a sort of rotational projection that just sweeps through the entire lattice. So we've projected 3D onto 2D to visualize it more easily. And what we can do with these resonances that I showed in the slide earlier is we can actually really locate these electronic defects to almost single sites in the diamond lattice. So you can see this x-axis here is nanometers. So we're really looking at sort of single atomic sites within the diamond lattice. And if you remember back to the numbers of what typical NMR experiments are looking at, this is a very different scale of magnitude in terms of number of spins that we're looking at here. So that's the story for electronic spins, which are slightly easier to detect than nuclear spins. So let me now move on to what do we do about nuclear spins. So both NMR and MRI are based on detecting nuclear spins, and they're a little bit trickier because their magnetic moment is more than 1,000 times weaker than the electronic moment. And so what do we do here? We will, we take our system. I've just seen there's a question here from Gareth Wilkinson, who says, does nanoscale imaging require use of a single crystal target? I'm not quite sure what that what this means. So the target, so one can use the target can be whatever one is interested in. So in the example that I showed just now, we were looking at defects inside the diamond, but what I'm going to show you now is using the diamond crystal to look at targets outside of the diamond. So the target, of course, is not a very useful sensor if all that you can sense is the spins inside the actual diamond, inside the actual sensor. So if I understood the question right, the answer is no, it doesn't have to be inside a single crystal. It can be anything. It can be in a liquid, and that's what we're going to have a look at now. It can be in a solid state system, or indeed it could be in a living cell. And again, we'll see some data on that in a couple of slides. So here for NMR, we're looking at a diamond nanocrystal here. So this is roughly 20 nanometers in diameter, and we've submerged it in a liquid of interest here, and we have the microwave antenna above, which is used, as I said earlier, to drive the spin, and the optics from above here. And here we're looking at, we have this sort of test material of fumblin oil, which has fluorine-19 spins, which is very nice because it's a very easily detectable, distinguishable spin species. So we have these fluorine spins, and then we also know that we have a thin layer of proton spins around this crystal, and that just happens naturally as these diamonds are exposed to the environment. So this is our system. And now if we probe this single spin using our quantum optics microscope, we can indeed see resonances. So here you can see the fluorine-19 signal and then the proton signal from the layer that's surrounding the diamond crystal. And we can change the magnetic field. Here you can see we're going from 24 millitesla to 30 millitesla, and indeed these resonances of the spins change accordingly. And we can put some numbers on this detection. So here we're looking at a detection volume of 19 nanometers cubed. So this is really truly nanoscale sensing. And we're looking at a concentration sensitivity of 270 spins per cubic nanometer per second. So we're really sensitive to quite a small number of spins. And indeed, the data set showed here on the left is for an NMR signal resulting from around 10,000 proton spins and fluorine spins. So this, again, comparing to the NMR numbers of billions of spins, this is still many spins, but it's a much smaller number of spins, and the key thing is that it's really localized to the nanoscale surroundings of this little diamond nanocrystal. And then, of course, we're not that interested in oil, really. We would like to do experiments maybe in living cells or other kinds of materials. And so here is an example of diamond nanocrystals inside lung cancer cells. So you can see maybe just about these yellow spots inside cells, which are clusters of diamond nanocrystals. And we've been doing some experiments in collaboration with a group here in physics and chemical engineering to make sure that the cells don't mind the presence of the nanodiamonds. So basically what we do to get the nanodiamonds inside the cells is we just introduce the nanodiamonds to the cell medium, and then they are endocytosed, so they're sort of eaten up by the cells. And we can use a detector for stress levels inside a cell that's dye-based to tell us whether the cells are happy or whether they feel really disturbed by having digested some diamond nanocrystals. And you can see here on the left that if we take these nanodiamonds as they come to us from the company that we buy them from, they typically have a graphitic shell, and that's not very nice. So graphite is quite reactive, and you can see that it's unpleasant to the cell because in the first hour this sort of stress indicator shoots up, and it does come down in the second hour, but really that was a rather disturbing event for the cell. Whereas if we do some preparation to these nanoparticles, if we oxidize them first and we get rid of this graphite so that it's really pure diamond, which is very inert as a material, then we don't see that stress spike, and we can be confident that we can introduce these sensors without perturbing the system, and we can actually do measurements in situ using these sensors. And here another example of some recent work here in collaboration with another group in physics is a confocal image of a bone cancer cell. And here again, you can see the nanodiamond fluorescence here around the nucleus and the cell autofluorescence lighting up in green. And in this project, actually, we're interested in looking at the temperature. So I've talked a lot about NMR linked to the results that we had earlier, but actually this particular quantum sensor can be used to detect other things as well. It can detect pressure, and it can detect electric field as well, and temperature. And temperature is a particularly interesting one for living systems. And so here on the right, you can see some experimental data on resolving temperature changes using these quantum sensors. And again, we're looking at temperature on the nanometer scale. So this is a rather different kind of tool to what typical thermometry would look like. And at the moment, we can resolve sort of below, well below 1 Kelvin or 1 degree Celsius within 1 minute. And I should say this is also part of a collaboration with the biochemistry department, where we're interested in looking at how the temperature rise in the oceans affects the temperature on the nanoscale in a symbiotic relationship between single cell algae, so dinoflagellates that are single cell algae, and their symbiotic relationship with corals. And these nanodiamond sensors will hopefully allow us to see what's going on in terms of temperature changes as the environment temperature rises. So that was most of what I wanted to tell you about nanoscale NMR. I want to finish by telling you a little bit about what we're thinking about next. And taking a slight step back, we can think about quantum objects interacting with each other and trying to understand sort of mesoscopic complex quantum ensembles. So let me illustrate this with a picture here. So on the left here, you can see these two dots, and you can assume that they're some quantum mechanical objects that have some properties such as spins or polarization or something like that. And they are very easy to model, even analytically, or to simulate. We know very well that they obey the quantum mechanics that we've known for decades. So this is the sort of very few particle or few object regime, which is very much quantum. We see quantum coherence, these kinds of superposition states that are short-lived but very interesting. And then on the other hand, there's this kind of infinite limit, where here I've got a big chunk of silicon, which still is quantum. As I mentioned, semiconductors, their behavior, the conductance behavior relies on quantum mechanics. But there isn't really any quantum coherence as such that's important to their behavior. And we can use so-called mean field theories that average over properties of particles to understand them really well. So both of these extremes are quite well understood. There aren't that many open questions, but of course here in the middle, there is this whole region of looking at many quantum objects that are not enough that you can just use sort of averaging theories, but they're still complex enough that you can't just run a simulation that takes into account every single degree of freedom in the system. And so, for example, here you get these exciting structures where you can, materials growers nowadays can generate these amazing materials that are different types of atoms layered on top of each other, and interesting sort of emergent phases occur at their interface that are very hard to predict, that we measure in bulk, large scale samples, but we don't really understand where they come from, just simply because it's complex enough that we can't really simulate it. So there are a lot of open questions sort of in this middle range. And this is this kind of collective quantum behavior where we do see quantum coherence effects without the averaging. And here the sort of keywords are many body localization, where we're thinking of a quantum state that sorry, just go. Many body localization, where we have a quantum state that typically if you take a quantum state and you let it interact with its environment, it will dephase. It will lose its information very quickly because these states are so fragile. But under certain circumstances, depending on the interaction that you have, these states might not decay. So you might get a very complex quantum coherent state that lives on for a very long time, and this is very unusual, and we don't quite know what governs this property. And of course, it's very exciting from a technological point of view as well. If you're thinking of realizing some kind of quantum technologies, if you can actually harness this quantumness for a long time and it doesn't dissipate into the environment, that's very exciting. And the other example is these quantum many body scars, where again, depending on the sort of initial parameters of the system, in one particular state of the system, so depending on how you arrange your spins, for example, the system might thermalize immediately, but at a slightly different initial state, you might get a very long-lived quantum state that can do all sorts of interacting, all sorts of interesting, that will live for long enough to show entanglement behavior and to show coherent evolution. So these are the kinds of questions that we would like to explore. And it turns out that actually these spins in diamond are a very nice testbed, or very nice system to realize these questions. So I sort of touched a little bit on these concepts. So here we're thinking of quantum simulators and quantum sensors. So as a quantum simulator is really a system that's simulating particular interactions that we're interested in. So this would be something that is too complex for us to just simulate on our computers, that we have to build a quantum system, say with interacting spins, for example, where we can tune and tailor their interactions so that the resulting evolution, the resulting time evolution, is what we're interested in seeing. And so here in Diamond, because it's such a well-controlled system, we have some recent results from last year where we were able to really tune the interactions. So you have some kind of spins that are interacting with each other, and really you would like those interactions to look slightly different, for the length dependence to be slightly different, or for the time dependence to be slightly different, in order to find out what would happen in such a system if I let it evolve. Would it sort of stabilize? Would I see some kind of many-body physics? And the key to doing that kind of, we call it interaction engineering, really lies in being able to control the system really well. And this is possible in some diamond-based systems. And another option is looking at these 2D materials. So same idea, but using nuclear spins in these single atom thick materials that can be stacked on top of each other, where again we have interacting spins, and we can control their interactions to simulate systems that we don't know yet how they're going to behave. And, and then there's the quantum sensor side of things that we spoke about earlier. And here again, we can try and look at this sort of mesoscopic scale of quantum interactions, where we have, for instance, here on the left, we're looking at a diamond sensor, which is not just static or inside a cell, but we can actually attach one of these to the tip of a scanning probe, such as an atomic force microscope, which is just a probe, like a tip that scans, raster scans over a sample of interest, and as it passes over the regions, it reports back on the topography. But in our case, if we have this diamond spin on it, we can actually also get information about the magnetic properties, so some kind of spin texture, some kind of underlying spin texture of this material, and we can also get we can also get information about conductance properties, for instance, investigating superconductivity and magnetism. And again, because this is so high spatial resolution, really on the nanoscale, we can start to see these kinds of phase transitions, so where the material starts behaving in completely different ways depending on the external parameters on the nanoscale, which is really key to some of these new phenomena that are predicted in novel materials. All right, so this is a little bit of an outlook, and I'd like to finish by thanking all these people. So of course this is a team effort, and there are a lot of people involved in the experiments that I showed earlier. So here are the students and postdocs who worked on the NMR project and on the cell work, and a few PIs who are collaborating on this project, all in Cambridge, from the physics department, Cancer Research UK, and biochemistry. All right, so I would like to thank you all for your attention, and open the floor for any questions that you may have. Thank you very much. Okay, so we have a question from Gareth Wilkinson. A couple of questions, actually. So let's see. What could be the potential applications of nanoscale collective quantum behavior? So there are a number of potential applications. So if you're really thinking of sort of applications in terms of what device can I hold in my hand in 10 years that will be based on this, of course, it's a little bit hard to predict, but so one exciting prospect is quantum memories, for instance. So there are, in emerging quantum technologies, there's a lot of emphasis on quantum communication, so photons transferring information from one physical location to another. But of course, once you get there, you need to store the information somehow. And you can't just store it because there is sort of quantum coherence and phase information in there as well. You can't just sort of store it on a piece of paper or in a classical computer. You need another quantum object to store it in. And so if you can build a system that's sort of robust on that scale, that can store complex states for a long period of time, that's a really exciting application. And I think in terms of really in terms of simulations, there are also many potential applications. So trying to simulate ground states of molecules, for instance. Again, if we can control these interactions, and we can mimic, basically the idea is that you have a particular molecule that has some kind of energy terms, some kind of Hamiltonian, and if we can now make a similar system that mimics those same energy scales, looks completely different in a physical realization, but mimics those same energies, we can find out more about interactions in molecules, in single molecules. And these are calculations that are much more too complicated for our computers to perform currently. And yes, this is related to quantum computation. So of course, again, the whole storage and communication is all linked. Okay, we have a couple of other questions that I will move on to. Let me have a quick read. Okay, so a question from Denise saying, how large irradiation intensity would be necessary to measure an external electronic spin? And could the necessary irradiation intensity potentially destroy biomolecules of interest? Thanks, yeah, that's a great question. So I presume you mean the optical irradiation intensity. So currently we use quite low intensities. So this is tens to hundreds of microwatts at the site of the defect, and I think this is on the order or even lower than a lot of the typical super resolution imaging techniques or confocal imaging techniques that are used. So because it's a single defect, it doesn't actually require very high intensities of light to be excited. We're really just relying on this one single spin to be excited and to decay down to give us information about the cell. So of course it's something that we do need to think about also in terms of interactions with other dyes, for instance, that might be used to label cells, whether there's any kind of crosstalk there, but we haven't seen any issues there. And for instance, so there have been experiments, not in our labs, but there are experiments using these NV centers to detect one single electronic spin that's external to the diamond, and that's possible. I guess it all depends on what kind of electronic spin is it, how strongly is it bound to wherever it is. Can it be easily excited? But yeah, basically the energy levels are not very high and not higher than other optical techniques. So then we have another question from Xiuyun Liu, asking, is it possible to use a quantum sensor to detect blood flow in the brain? The skull is a rigid box and traditional detection technology cannot penetrate it easily, except X-ray or MRI, etc. So yes, indeed it is. And there are some research directions, so there are people who are trying to do this. I haven't heard about the brain, but certainly blood flow in the heart There's a project in Warwick, actually, where a research group is looking into that. And here, yeah, there you're not making use of the sort of nanoscale resolution that this sensor gives you, unless you really want to inject diamond into someone's brain, which is not completely unthinkable. I think there have been experiments of injecting nanodiamonds into mice and they didn't seem to mind, but perhaps this is not immediately what one would want to do. So typically for the blood flow measurements, it would be taking a bulk diamond, a millimeter-sized chip that you could then stick to close to the brain, and then it depends on how close the blood vessels are, whether that would be meaningful or not. But I haven't thought through the numbers on that yet. Okay, thanks. Then we have a question from Keith Mansfield saying, I'm going to read it. If we have, so at the beginning you showed an MRI of the brain at millimeter scale. If we have nanoscale MRIs of the brain in the future, might this pave the way to whole brain emulation and the possibility of either storing our brain states, if non-destructive, or scanning them for upload, if destructive, but we're dead/dying? With brackets, ignore if this is too wacky a thought. Okay. That's an interesting thought. So I think if you're going to cut open a brain after someone's died to sort of map it out and store it for posterity, then possibly this is not the right technique, because you would be, there are other ways of looking at structure inside the brain that will probably be better resolution if you're going to kill the thing anyway. And then for live, again, that depends on whether you're happy for diamonds to be introduced into your brain. And I mean, there is a group at Harvard that's used diamond sensing to look at neural signals. So not in humans, but in a worm. So they had a worm strapped onto a diamond plate. It's probably not a very happy worm. And then they were triggering electrical signals from one end to the other. And they were able to measure this using the diamond sensor. So that's possible in principle, but I think I'm not sure we'd be able to map out the entire brain structure using this technique. So then there's a question from Stephen Potter. Quantum computing demands ultra-low temperatures. Can the isolated spin states in crystals mean that quantum computing could be carried out at room temperature? So for quantum computing, it's you could build a rather bad quantum computer out of these defects at room temperature. So there are really nice experiments using these NV centers as sort of quantum registers, as sort of memory storage, quantum memories where they interact with nuclear spins that are in the diamond lattice surrounding them. So it's kind of an array of quantum objects interacting with each other, and this is going towards the quantum communication, quantum computing route. But these experiments are done at low temperatures, and the reason for that is that the optical interface is much better. So in a lot of these applications, you want something that's local that can do quantum computations, but then you need to extract the information somehow, and you want it to communicate with a different quantum computer somewhere else. And so in order to do that communication, you want an interface to photons. That so far that's the most efficient way of transferring quantum information for long distances. And if you go to long temperatures, actually, so I showed the emission profile from NV centers. So I said we detect red photons. Of course, they're not just at one frequency. There are a number of frequencies, and it turns out that at room temperature, actually, because of the lattice vibrations, you get a whole spectrum that's very wide, which doesn't matter if you're just trying to read out the spin state, which is what we do. So the entire broad spectrum modulates depending on the spin states, so we can use that. But if you're trying to extract a single photon that carries the information from your NV center, then having that photon potentially distributed over a large range of frequencies is not a helpful thing. So people using these NV centers for sort of quantum communication, quantum computing applications do actually cool them down to not millikelvin temperatures, which is sort of the coldest, at least commercially available systems that you can get, which is typically what the superconducting qubit quantum computers are based on, but at least to sort of a few Kelvin, which is still quite expensive. So unfortunately, I think no quantum computer, or very, very bad quantum computers at room temperature are possible using this system. So then there's a question from Paul Jarman. Does the technique work with materials other than diamond? So yes, actually there are. So first of all, there are different kinds of defects in diamond with which this works, and then there are also, for instance, silicon carbide, which looks very much like diamond. So the lattice is pretty much the same. It's a diamond lattice structure. But every other atom is a silicon atom. And so there are similar kinds of defects that do similar things. And they have slightly different properties. And basically, in terms of quantum sensing, nothing has really been able to beat these NV centers in diamond, but people are still searching. And this is really an active field of research to try and find good defects in other materials that can perform similar tasks. So, a couple more questions. So there's one from an anonymous attendee. What do you hope to discover using this research in the future? I think the thing about sensors is that if you have a really good sensor that has unprecedented properties, that has this spatial resolution that can't really be matched together with the non-invasiveness, there are many options. So I think anyone who's working in the field, you ask them, they'd probably give you a different answer. So I think I'm really interested in sort of nanoscale thermodynamics and quantum many-body physics. So the kind of things that I touched on at the end. So that's kind of one side. So I would really like to be able to see some evidence for these out of the ordinary many-body localized states, for instance, and understand sort of thermodynamics on the nanometer scale. So there are a lot of theories that really apply to sort of averaged quantities. So taking some kind of mean field approximations, and then when you go to the nanometer scale, everything changes, and you get very strange effects. So I would like to explore that more. Yeah, really see these, there have been many predictions of these long-lived quantum states, especially in two-dimensional systems. I would really like to see that. And then on the other hand, I would also really like to apply this to different systems. So I'm really excited by the biology work that we're doing, that I showed on the dinoflagellates system. So really being able to perform temperature sensing and these nanoscale MRI measurements on the nanometer scale we have a project looking at cancer cells, for instance, as I showed earlier. And again, being able to, because it's such a well-controlled system, you can imagine taking cancer cells or cells that can be triggered to become cancerous. And during this process, you can watch them using these defects. So you can say, oh, actually the temperature's risen a little bit around the nucleus at this point in time, and that's what triggers a whole set of other problems that then leads to the cancer actually developing. So it's really exploring how things change on the nanoscale, because this is a regime that we haven't been able to access until now. And there are some other fun questions, like there are magnetic inclusions in meteorites that can tell us something about the Earth's magnetic field millions of years ago. That again, these are magnetic inclusions that are on the nanometer scale, so they're very hard to image. So there's no sort of quantum physics questions there, but I think it's a really exciting tool to explore some of these open questions in very different areas. Let me see, there are a few more. Stephen Potter is asking, can there be any similar defects in 2D lattices, such as graphene? So yes, there are defects in 2D materials, such as graphene and these TMDs, another family of these one atom thick materials. They don't have these kinds of properties, though. They're typically the sort of magical thing about this NV center is that you can use it at room temperature, which is not really the case for these defects in other materials or in these 2D lattices, and that you can do this purely optical spin state readout, this kind of decay pathway, which either gives you the red glow or doesn't give you the red glow. So that's very unusual. So yes, there are many defects in other 2D materials, but for these defects, you have to take them down to very low temperatures to be able to do useful things with them. And typically these are used more for sort of quantum communication applications rather than this kind of room temperature sensing. So let me see. So there's a question on applications to power generation from Mayoma Onwoche. Do you see any applications to power generation or carbon removal technologies? I think the short answer is no. There are some, I mean, this is probably not what you mean by power generation, but there are some related experiments in these kinds of systems looking at sort of heat machines and sort of in a sense power generators, but on the nanoscale. But this is really with an aim to understanding thermodynamics in the quantum world. So I think this is not as a practical an application as you would like to see. And maybe I'll take one more question. There's a question on the temperature sensing, how that works and how that relates to the MRI detection from an anonymous user. So this works in a very similar way in terms of the detection. So the optics and the microcontrol is the same, and the thing that gives you the signal is not, in the case of the MRI, where we have these energy levels and they shift apart as we bring a magnetic field close by, but for the temperature, the diamond lattice actually physically expands and contracts, and this expansion and contraction changes the electronic orbitals and changes the energy levels, and so they shift in a slightly different way. So instead of shifting apart like this, they shift both in parallel up and down, so we can distinguish between the two. So when we're doing experiments, say with living cells, we can distinguish a temperature change from an MRI signal. So that's the physics behind the temperature generation. Okay, I think that I can see there are quite a number of more questions, but I think we're going to have to close this session down now. So I would like to thank you all for tuning in today and for listening. I hope you enjoyed this talk, and thanks especially to everyone who asked all these great questions at the end. And this is, I think, the first day of the Alumni Festival, and there are many more events to come in the next days, and I hope you enjoyed this talk and you learnt a little bit about quantum sensing and nanoscale MRI. Thank you very much.